All right, everybody, welcome back to the deep dive. Today we're diving into some really fascinating science. This is an area called Xenotopic synthetic biology. Have you heard of this?
I have.
Yeah.
I have to admit, when I first heard this term, I was a little intimidated just by the name itself. Xenotopic synthetic biology.
It's a mouthful, right?
It's a real mouthful.
Yeah.
But actually, when you break it down, it's a pretty simple and incredibly clever concept.
Yeah, and it has huge implications, particularly when we think about aging and the diseases that tend to kind of, come along with aging. And in fact, that's really what we're going to be focusing on today as we kind of go through this deep dive on this topic. Please don't forget to like the video and subscribe if you want more content like this. So xenotopic synthetic biology, maybe we can break that down a little bit. What is that really all about?
Yeah, so I think the really intriguing thing here is that it represents a fundamental shift in approach.
Mm-hmm.
Right. So instead of just looking at our own genes and enzymes to solve the problems of aging, this field really explores the vast biological resources that are available in other species. Right?
Oh, interesting.
It's like we're expanding our toolkit in a really radical way.
So it's like we're looking at our own systems and thinking, okay, how can we tweak our systems to, kind of address these issues? But it's like, well, maybe there are answers outside of our own biology.
Precisely.
That could be really interesting to look at.
Yeah, and, our biological systems are amazing, right? They're the result of millions of years of evolution.
Yeah.
But other organisms, they've evolved their own unique solutions to similar kinds of biological challenges. And xenotopic synthetic biology is all about identifying those unique solutions in other creatures.
Right.
And seeing if we can adapt them to address the metabolic issues that drive aging in us.
I see. So we're really going outside of our own systems, looking at nature and being like, well, what has nature come up with that we can kind of borrow?
Exactly. It's like borrowing from nature's toolbox.
Ooh, that's a good one.
Right.
I like that.
Finding those unique tools that our bodies just don't naturally possess.
That's cool. So this isn't like a completely new idea that just popped up overnight, is it?
No, not at all.
Okay.
The concept of using biological tools from one species to affect another, this has roots going back decades.
Okay. So do we have any early examples of this that we could talk about?
Absolutely. So there's a fascinating story from the late 1940s.
Okay.
A researcher named Dr. John Kidd.
Okay.
He noticed that something in guinea pig blood.
Oh, wow.
Could actually stop lymphoma from growing in mice. And it turned out that this something was an enzyme called asparaginase.
Interesting. So that's like guinea pig blood stopping cancer in mice.
Exactly. And that discovery was a real turning point, right?
Okay.
It showed that an enzyme from one mammal could have this powerful therapeutic effect in another. And suggested that the kind of biological solutions found in nature, they could transcend species boundaries. And it was an early hint that our own genomes might not hold all the answers to our biological problems.
Right. So this is like 70 years ago that they were finding these things. It's not just like a brand new field.
Not at all. And it wasn't just a fluke either.
Oh, okay.
Later on in the 1960s, scientists discovered another enzyme called methionine. And by the 1980s, this enzyme was being tested as a cancer treatment in rats.
Okay. Interesting. So these are like enzymes that already exist in nature that they're finding, and then they're like, oh, this could be useful in this situation.
Exactly. So the idea of borrowing these biological functions from other organisms, it was gaining traction in different contexts.
Right. Not just for, aging, but for other things as well.
Precisely. And these examples, while focused on diseases like cancer, right, they hinted at a broader principle that we could potentially leverage the unique biochemistry of other organisms to tackle these fundamental biological processes in ourselves.
That's cool. And so how did that idea kind of progress?
So fast forward to the late 1990s.
Okay.
And we see this really elegant piece of research from Dr. Takao Yagi's lab. They took an enzyme called NDI1 from yeast.
Oh, wow.
And they introduced it into hamster cells that had a defect in their mitochondria, the powerhouses of the cell.
Right.
And these hamster cells, they were really struggling to produce energy.
Okay.
But the yeast enzyme, it actually stepped in and restored that lost function.
Oh, wow. So they took an enzyme from yeast, put it in hamster cells to fix a problem that was in the mitochondria.
Exactly. And what's truly significant about that experiment is that it demonstrated that we're not necessarily limited by the evolutionary constraints of our own genome.
Yeah.
When it comes to addressing these fundamental cellular deficits that contribute to aging.
Right.
It opened the door to the idea of these cross-species functional replacements at a very basic level.
Okay, so how did this progress into the 2000s then?
So building on that, in the 2000s, you saw some exciting developments
Okay.
with both that yeast NDI1 enzyme that we talked about and another one called AOX from sea squirts
Okay.
being tested in living organisms. And researchers were starting to explore their potential not just to treat these specific diseases, but to actually influence the aging process itself.
Oh, wow. So actually directly targeting aging.
Exactly.
That's really cool. So this is a field with like a really solid foundation of discovery then.
Yeah, it's not just some kind of pie-in-the-sky idea.
Okay, cool. So why is this xenotopic approach potentially more advantageous than just trying to manipulate our own genes and enzymes when we're thinking about tackling aging?
That's a great question. So the core promise here
Okay.
is that these borrowed enzymes, they might offer completely novel ways to interact with the incredibly complex metabolic pathways that are so tightly linked to aging.
Okay. So they might be able to do things.
They might be capable of performing tasks or achieving efficiencies that our own evolved systems just aren't equipped for.
I see.
And that could potentially lead to more effective ways to intervene in the aging process.
So they can target things that our systems just like can't?
In a way, yes. Think of it like having access to this vastly expanded set of tools, right? Our own enzymes, they've evolved within a specific biological context. But by looking across the entire spectrum of life, we might discover enzymes with these unique properties or catalytic efficiencies.
Right.
That could be incredibly beneficial when introduced into our systems to address specific age-related metabolic dysfunctions.
Okay. And so to get a better handle on this, it's really helpful to think about these xenotopic enzymes in terms of like the specific metabolic areas that they target.
Right.
It gives us a kind of framework for understanding their potential impact on aging.
Absolutely. So what are some of the key metabolic parameters that these xenotopic enzymes are designed to influence?
Okay, well, let's dive into some of the key areas that are highlighted in our sources. First up, we have enzymes that play a role in amino acid metabolism.
Okay.
Things like regulating the levels of specific amino acids, like methionine, asparagine, and arginine within our cells.
So we've already touched on methionines and asparaginase.
Right.
What's the underlying rationale for focusing on these particular amino acids in the context of aging?
Well, there's growing evidence from research in various model organisms that restricting the intake of certain amino acids, most notably methionine, can actually lead to an extension of lifespan and improvements in health span. So the idea is that xenotopic enzymes that can degrade these specific amino acids could potentially mimic those beneficial effects of dietary restriction without requiring drastic changes to what we eat.
I see.
It's a more targeted biological approach to achieving a similar metabolic outcome.
That's a key insight. It's about achieving this specific beneficial metabolic state through this precise enzymatic action rather than relying solely on these broad dietary interventions, which can have other complex effects.
Yeah, it's a very specific tool for a very specific job.
Right.
What's the next major category of influence?
So the next really significant area is mitochondrial function.
Okay.
We've already mentioned those yeast and sea squirt enzymes, NDI1 and AOX,
Right.
which can essentially bypass damaged components of the mitochondria's energy production machinery.
Right.
But there are also these other fascinating tools in this category, like MTON and MitoDR, which are these light-activated proton pumps that can actually boost the electrochemical gradient across the mitochondrial membrane,
Wow.
essentially supercharging their energy production potential.
So like giving the mitochondria an extra boost.
Exactly. And considering the central role of mitochondria as the energy hubs of our cells, right? And how mitochondrial dysfunction is a hallmark of aging. It's not surprising that this is a major area of focus.
Yeah, for sure.
The ability to bypass these deficits or even enhance mitochondrial activity using these borrowed enzymes could have these really profound anti-aging effects at a cellular level.
Totally. Then we have a really interesting group of enzymes that can modulate the cell's redox state.
Okay.
Think of the redox state like the balance of electrical charge within a cell, which is crucial for all sorts of processes. This involves things like the ratios of important molecules like NADPH and ADP plus, NADH and AD plus, and CoQH to CoQ. Enzymes like LBNOX, TPNOX, and ECSTH fall into this category.
So the redox state is absolutely critical for this vast array of cellular functions, right?
Yeah.
From energy metabolism and signaling to our defenses against damaging free radicals. And precisely fine-tuning these delicate balances with xenotopic enzymes could have these really far-reaching and beneficial consequences for the aging process as a whole.
Wow, so that's a really important area to be looking at then.
Absolutely.
What else?
Another intriguing area
Okay.
involves enzymes that directly impact the ratio of lactate to pyruvate, which are these key intermediate molecules in how our cells produce energy from glucose.
Right.
LOXCAT and GRUBRA are examples of enzymes that can influence this balance.
And so why is that an important thing to manipulate?
Well, manipulating that lactate-pyruvate ratio.
Okay.
It can have these significant effects on everything from glucose metabolism to cellular signaling pathways. It represents another potential lever we can pull using these external biological tools to influence the trajectory of cellular aging.
I see. Okay, cool. And finally, we have a set of enzymes that target glutathione levels, that's GSH, and the production of reactive oxygen species, which are often referred to as free radicals.
Yeah.
Enzymes like STGSH, DAAO, and killer-red SOD1 are involved in managing oxidative stress, which has long been implicated as a major driver of aging at the cellular level.
So while our understanding of the free radical theory of aging has become more nuanced over time, there's no question that maintaining that healthy balance of antioxidants and managing excessive oxidative stress is crucial for cellular health and longevity.
Right.
And these xenotopic enzymes, they offer these very specific and targeted ways to modulate those ROS levels and bolster our antioxidant defenses.
It's like we have all these tools available to us now. That's really cool. So as you can see, the field of xenotopic synthetic biology is incredibly diverse, offering a really wide range of potential tools that can target these fundamental metabolic shifts associated with aging and age-related diseases.
Right. It's all about looking beyond the limitations of our own biology and harnessing that vast biochemical innovation that nature has already produced in other species.
Right. It's like looking at nature and being like, well, you've already done the work.
Exactly.
I'm just going to borrow that from you.
That's a great way to put it.
What's particularly exciting is the potential for using these xenotopic enzymes in combination, right?
Absolutely. The sources highlight that combining enzymes that target different but interconnected pathways could lead to these additive or even synergistic effects in promoting health span and extending lifespan.
So it's not just one thing, it's like maybe we can use multiple things together.
Exactly. It's a move towards these more sophisticated and potentially more effective interventions.
And so that's like a really hopeful sign.
Absolutely.
That maybe we can really, come up with some solutions here.
It suggests a move away from this one-size-fits-all approach to aging interventions and towards these potentially very targeted and personalized strategies for promoting healthy aging based on an individual's specific metabolic profile.
Wow, that's really cool. Okay, so to bring it all together, the key takeaway from this deep dive is that xenotopic synthetic biology offers this truly innovative and potentially powerful strategy for tackling this complex challenge of aging and age-related diseases.
Right.
And it achieves this by intelligently and strategically leveraging the unique biochemical capabilities that have evolved in other species.
If we zoom out and look at the bigger picture, imagine a future where we can routinely use enzymes sourced from a variety of organisms, from simple yeast and bacteria all the way to other mammals, to precisely fine-tune our own internal metabolism. This could potentially delay the onset of age-related decline and offer these completely novel and more effective ways to prevent and treat a wide range of diseases.
So given all of this, the incredible diversity of these metabolic pathways that these xenotopic tools can influence, from the basic building blocks of amino acids to the intricate workings of our mitochondrial powerhouses and the delicate balance of our cellular redox state. What single area do you find most promising or intriguing for future research aimed at extending our healthy years?
That's a big question. It's a question that really underscores the transformative potential of this field.
It certainly does. It really makes you think about what's possible.
It raises some really profound questions for all of us as we contemplate the future of research into aging and longevity.
Well, that brings us to the end of today's deep dive into xenotopic synthetic biology. This is a field that's rapidly evolving.
Absolutely.
And it's brimming with potential, and we've only just touched on some of the key concepts and examples.
There is indeed a vast amount more to explore in this fascinating intersection of synthetic biology and the quest for healthy aging.
Please don't forget to like the video and subscribe if you want more content like this.